Welcome to part 2 of my lecture on chapter 11. We're still going to continue talking about how gene expression is controlled. Interestingly, only about 1.5% of the human genome actually codes for proteins. The same thing is true for a lot of other organisms as well. And then you have another small percentage of the DNA that contains genes that code for ribosomal RNA or rRNA and transfer RNA or tRNA. For a long time, it was thought that a lot of the other parts of the DNA was just junk or nonsense. They called it junk DNA because scientists could not figure out what the function of those sequences was. But there has been a lot of recent information that came from scientific observation and experimentation that has shed some light on the function of these junk DNA sequences. And so a lot of the genome is actually transcribed into functioning RNA that does not code for proteins. Researchers have discovered evidence of the actual function or biological roles of these RNA sequences. And in '93, researchers figured out that there are some small single-stranded RNA molecules called MicroRNAs or miRNAs. And these miRNAs have the function of binding to complementary sequences of messenger RNA molecules. And the microRNA strands are about 22 nucleotides long, so they're tiny, and they bind to proteins, forming these microRNA protein complexes, which are able to bind to complementary mRNA molecules. And the complementary mRNA molecules need to have between 7 and 8 nucleotides that are complementary to the microRNA sequences. And if the mRNA molecule contains a sequence that is complementary to the entire microRNA molecule, then that protein microRNA complex has the function of breaking down the target messenger RNA molecule. So if the messenger RNA molecule only matches part of the microRNA sequence, then the complex keeps that particular strand of mRNA, from being translated into a polypeptide chain. And so based on this emerging body of data, biologists estimate that microRNAs can regulate the expression of at least half of human genes, which seems like a lot, considering that, just around 30 years ago, microRNAs were completely undiscovered. Another type of RNA sequences that have been discovered to have functions in the regulation of gene expression are called small interfering RNAs or siRNAs. And they're very similar to microRNAs. Both can form complexes with the same proteins and have similar impacts on gene expression. But there are some small differences. Let's see. So small interfering RNAs can block gene expression as well. And that mechanism is called RNA interference, or RNAi. And that is the mechanism through which small interfering RNAs block gene expression. And so scientists have been able to use small interfering RNAs in order to control gene expression. So it's actually, they have been able to take this information and apply it. One example is injecting siRNA into a cell and turning off the expression of a gene with a sequence that matches that siRNA, and being able to disable specific genes allows scientists to be able to take a look at the function of those genes. That's one way that scientists have been able to figure out what genes code for what proteins and what the functions of those are. So there have been a lot of subsequent discoveries and information that has been gained since the discovery of the small interfering RNAs. In 2006, there were two American scientists, Andrew Fire and Craig Mello, who were awarded a Nobel Prize for their discovery and categorization of RNA interference. For more information on this topic, you can read section 11.6 in your chapter. This checkpoint question is a good way to check what you've learned and also to remember how base pairing works. So if a gene has the sequence of AATTCG, then what would the sequence of an MI or microRNA be that turns that gene off? Hopefully you got the fact that the sequence listed here is a DNA sequence, so then you would have to figure out the complementary mRNA sequence to that, which would be UUAAGCGC, and so then the complementary microRNA sequence to that mRNA sequence would actually be AAUUCGCG. Hope that makes sense. Okay, so gene expression, and this is section 11.7. We're going to talk more about the different mechanisms that regulate gene expression in eukaryotes. Gene expression can be regulated in different ways in the nucleus and in the cytoplasm. And you can look at the figure that is section 11.7 in your ebook, and it kind of gives a visual summary of what we just talked about. It talks about the different mechanisms for gene expression regulation, and that is, again, figure 11.7 in your ebook. In section 11.8, we talk about how studying the development of fruit flies allowed and continues to allow scientists a chance to look at how genes are expressed in different body parts. And selective gene expression, which we talked about earlier in the chapter, is really important during the development of an embryo from a zygote, and different phases or waves of gene expression occur, and you have the products, which are proteins, of one set of genes activating other sets of genes, and that is common when you are in that stage of development when the embryo of a multicellular organism is developing from a zygote or from that first cell. And one concept I wanted to talk about is homeotic genes. And a homeotic gene is a control gene that has the function of regulating groups of genes that determine the anatomy of body parts. One example is which body parts will develop where in a fruit fly. And so you have homeotic genes in fruit flies that instruct cells in segments of the head and thorax, which is kind of the mid-body in between the abdomen and the head, to form antennae in the head, and then those segments of cells in the thorax form legs. All right, and there are various techniques that scientists can use to study how genes work together. Nucleic acid hybridization allows researchers to figure out which cell types express a specific target gene, and then a DNA microarray is a technique that can be used to obtain data about which genes are turned on or off in different cell types. And DNA microarrays, one potential application is that leukemia could be classified based on DNA microarrays, and then that classification into different subtypes would be based on the activity of 17 genes, and that would help for doctors to treat the patients based on the type or subtype of leukemia that they have. Another concept from section 11.10 that I wanted to touch base on is the signaling between cells that allows cellular activities in multicellular organisms to be coordinated so that the organism works together, or the cells of an organism work together, and proteins and other kinds of molecules can carry messages from the signaling cell to the receiving or target cell. And in most cases, a signal molecule works by binding to a receptor protein in the plasma membrane of the target cell. And signal transduction pathways are series of molecular changes that convert a signal on a target cell's surface into a response within the cell. Okay, so to turn on a gene, must a signal molecule actually enter a target cell? The answer is no, that the receptor protein on the cell membrane of the target cell would receive the signal, and then it would kick off a signal transduction pathway, which would lead to the changes or the response inside the cell to that particular signal. There are similarities amongst different types of organisms in terms of how their cell signaling systems work, which suggests that these signal transduction pathways evolved early in the history of organisms. This concludes part 2 of my chapter 11 lecture. I'm going to do one more lecture, and that is going to be talking about the cloning of plants and animals, as well as some of the medical applications of the information that we have talked about earlier in this chapter.